And oh well. And no,
maybe while people are arriving, you can tell me what the title means. Yupay, Amuyuni, Sarnaqaña, Yachay.
Oh, this is about the PDE, about the word. And what? This word is about the PDE in the about the clustering, about the machine learning, informatics, or what topic?
No, I mean the title of the colloquium.
Yeah.
No, what does it mean?
What does it mean? The meaning is about some people working with numbers ideas in English. About Yupay, Amuyuni, Sarnaqaña, Yachay is about Quechua with Aymara. Is native language of Peru and some countries with Bolivia, Chile. We talk together with native. With the Spanish, is about the programming language. Is the title of the project.
I really like that.
It's about the Quechua speaking or the Germany, I think, because in the PhD school in Cuba, some students with German, and the words is very similar to pronunciation probably. I don't know about your pronunciation with the same words. The Yupay, Amuyuni, Sarnaqaña, Yachay is Quechua and Maya words. Yeah. No? About 5 minutes waiting for more coming students. And start the talk. Now it's about 11 p.m. I think in Germany, right?
Yeah, that's right. So for me, this is an evening entertainment.
Oh, I very apologize for the
It's okay. We scheduled it this way because I didn't know if I would already be in the US or in Germany. And so I remember we picked the time so that I could do both US and Germany. So it's fine.
Oh yeah, thank you very much. And apologies if any comments for the time. I don't know, it's about the 7 hours of the difference or the time share. In what?
Yeah, no problem.
Yeah, it's about the waiting 5 minutes and start, miss. Wait a minute. Well, and now, Miss, now are you working some project about research on the biological mathematics?
I have so many projects. So I started to work in different directions. So yes, I still work on a project related to exactly this work actually that I'm presenting today. But I also started working in data clustering and data classification algorithms, and in particular using graph-based learning. There is something called the graph Laplacian, which is like a discrete diffusion operator that you can construct on a graph that you build out of data points. And it's like a discrete version of a diffusion operator. So what is really nice is that you can then, if you have a lot of data points, you can approximate the setup by a PDE, by an elliptic PDE. And so then you're back in PDE analysis. So I've been using some of these PDE techniques to analyze these clustering and classification algorithms.
Oh, I know. Now the PDE and the functional analysis with the complement about the PDE. It's not my strong topic about the PDE. I in some base very basically about the PDE. I work more in OD, DDE, and impulse equation. It's my strong topic, but I know about the PDE, about the Solev and Hilbert spaces. It's so hard for the work, but I know it's very important about the PDE and the notion in the work. We're coming now.
And yeah, then I also work now on some optimization and sampling algorithms. I did some project with material scientists, with computer scientists. Yeah, so it's very diverse what I'm doing. So it's not just this topic of biomathematics. But what you will see, I think, is that once you have understood sort of these the structures of the equation or the properties of the equation, then you can find these structures again and again in different application fields. And so, you know, a mathematician is kind of like a chameleon. If you understand the structure of the equation, it doesn't matter what the application is, right? You can still say something about the behavior of the problem.
Oh, I know about this stuff with the complement with the calculation. Well, it's about now in 1 minute. You can start the talk, and thank you very much for the accept to the this participation, this project. Thank you very much. You can talk about the 40 minutes approximately, about some 15 minutes for any question and consults. And well, and thank you very much again for accepting the talk here in this project about the scientific talks. And you can start, Miss.
Okay, thank you. Thanks a lot. Yeah, it's my first time giving a talk in Peru, even if it's just virtual. So I hope that one day I can come and visit. Yeah, so today I want to talk about chemotactic clustering. And yeah, this is a topic in biomathematics, and actually these kind of bacterial chemotaxis equations are a class of equations that many mathematicians have worked on over the past decades. And I want to summarize a bit what has been done there. But before I do that, I give a kind of very general introduction, because I heard that this colloquium is very diverse, and that we have people from various different fields. So this is why I thought I'd just start with a very basic question, sort of what are PDEs actually? So some of you might be experts on PDEs, of course, but some of you maybe are not so familiar with the analysis of partial differential equations. But what I like to say to people who have not worked with PDEs before is that they are just a way of describing change. So the way how I think about that notion of change is that this is really what characterizes most things in the world. It describes how things change. So motion is just a change in position over time. Growth is a change in size over time. So most of our observations or our knowledge about the world, we can actually put into physical descriptions of change. And so, as you know, derivatives are just rates of change, essentially, with respect to some quantity. And so then differential equations is just a relationship between these different derivatives for some given function. And so then when we solve this differential equation, we basically want to find the function itself just from knowing how things change, just from the rates of change and how they're related. And so I want to start with a very sort of simple example, but it's actually representative for a whole class of models that we will also meet later on in the talk. So the simplest thing I can think about is just a car that is traveling down a street. And what you want to measure is how far you have traveled. So what is the distance that you have traveled at some time when you started at time equal to 0. And so this is your unknown. But what you know is the velocity that you have traveled at. So let's say you were in your car and you always saw how the velocity changed on the way. And so then this is your very simple ODE that probably most of you know how to solve without any problem. So given this velocity function, you want to find this x of t. But now you can lift this up to many, many different particles. So here the particles are cars, so you would have then many, many cars. And if you wanted to describe, instead of describing just one car, you wanted to describe many cars, you can instead write down the evolution of the distribution of cars. So this rho of tx is sort of the probability that a car is at a certain position x at time t, or you can think of it as just a distribution of lots of cars. And then if each car evolves according to this little equation here, then the distribution of cars evolves according to this PDE. And so this is the continuity equation. And you see that here I have dt and I have dx. And so these are different partial derivatives. There's the rate of change with respect to time and rate of change with respect to space, right? So really partial derivatives just means that you look at the rates of change with respect to the different independent variables. And so the symbols that you will see frequently is sort of the gradient, which is just a vector of special derivatives, and then divergence. And also Laplacian, I didn't write it here, but the Laplacian, you just sum up the second derivatives in all the different directions. So the way how I think about PDEs is basically just giving us a relationship between the different rates of change of a quantity. And so these models that I want to talk about is a very specific class of PDE models that describe how cells move. And so to that, let me give you just a little introduction to what bacterial chemotaxis is. So chemotaxis just means that cells are able to sense chemical signals, so chemical changes in their environment. And a very typical type of bacteria that is able to do that is the E. coli cell, Escherichia coli. And this type of cell is actually quite common. So most of us, we carry it in our digestive system as well. And it's completely harmless most of the time. There are different strains, and some of them are poisonous also. But the reason why it's we know a lot about it, and so it's easier to model for us, is because first of all, it's easy to reproduce in a lab environment. There have been a lot of studies done on E. coli cells. And then also they're sometimes regarded as representative for other types of cells. So you might study E. coli cells and then infer behavior for a bigger class of cells. So this is why there has been a lot of modeling done for E. coli cells. And so to write down a model of how a whole group of cells behaves together, we have to understand what makes them move. So they move around based on some kind of communication, right? But how do they communicate, right? Because it's just, they're single cell organisms. So they don't have any eyes or ears. Like, how do they even know what's going on? So actually, there's two things. So, well, there's more, but let's, to keep it simple, I will focus on these two. So they're able to sense chemical substances, sometimes several different chemical substances. And then they're also able to produce a chemical substance themselves. And so by measuring sort of the changes in concentration of the chemical that they're producing themselves, they get an idea of where the other cells are. So if they're close to the group or if they're very far away from everyone else. And then, okay, why do they even move, right? So there's sort of three main forces, if you want to simplify things, that make a cell move somewhere. One is just diffusion, so just a random walk. If you just drop a cell somewhere, it's going to move around, and that's a kind of diffusion process. Then they would like to eat, so they're trying to find a food source, so they want to consume nutrients. And then third, they want to stay together in a group. And they do all of these things at the same time, right? So what that means is that there's a kind of competition between different forces happening here. And in particular, so the random walk gives you a diffusion force, which you can put into a mathematical model. Then the chemotractant, I'm going to call the chemical substance that is produced by the cells themselves. And then you have food or some kind of nutrient that the cells are attracted to. And so this can become quite complicated as a process between these different competing forces. And so here I want to show you a model that has become very famous for describing the evolution of E. coli cells. And that's the Patlak-Keller-Segel model. So here you have three different quantities that you're modeling. One is the density of cells, of course, that you're interested in. So that's the rho of Tx. Then you have the concentration of the chemotractant, of the substance that the cells produce, and we will call that S. And then you have a nutrient concentration. And the X here, so the space variable, it could be in 1 dimension or 2 dimension or 3 dimension. So usually it's 3 dimension, right? Because we live in a 3-dimensional world. But you can also think of cells on a petri dish, for example. So that would be 2-dimensional. Or you can put them into a tube, and then you can measure 1-dimensional movement, right? And so this is this model. And I want to say a little bit about it. And then you see a lot of parameters here. So these are different constants. And these constants can be measured or estimated experimentally as well. So first of all of these three different quantities, they diffuse. And they diffuse with a different speed. So this d rho, ds, and dn, they are just diffusion coefficients. So they're constants, but they're different diffusion coefficients. And then let me describe this other term here. So if I start from the nutrient on the bottom, this gamma here is a consumption rate. So the food, the N, is being eaten up by the cells. And the speed at which the N decreases in time based on the consumption by the cells is this term here. So it's the gamma times rho times N. So if you have more cells, then the nutrient disappears faster, right? And this is evaluated at the point T and X, of course. Then the second equation, so the chemotractant, it says, okay, it also diffuses. And then this alpha here is the degradation rate. So this is basically saying that the chemical is reacting and is becoming something else in the process, so that the cells are not able to sense the chemical anymore. So you're taking this out of the equation. And then this beta here is very important. So this is the production rates, because as I said, the S itself is produced by the cells. So this is why there's new S, so S increases with this production rate beta based on the number of cells that you have at this point. And finally, for the equation for the cells, the really important term is this one here. So this is a drift term. This looks a bit like the continuity equation that I showed you in the car example. But now the velocity is actually the sum of the gradient of S and the gradient of N. And what that means essentially is that the cells are swimming up the gradients of the chemotractant and the food. So they want to go into the directions where you have higher S and higher N. So they measure the gradient, and if the gradient is positive, this is where they go, right? So this is, or if not, if the gradient is positive, but they go into the direction of the steepest gradient, basically. And you see that this term here makes these equation coupled. I mean, they're also coupled because the rho appears here in the equation for the S, and the rho appears in the equation for the N. And then both the S and the N appear in the equation for the rho. So if you want to solve this, you need to solve all of these three PDEs at the same time. And that's actually quite difficult to do. So there has been a lot of analysis done on this equation, but I want to show you a special case of this equation that has received particular interest by mathematicians, and I will explain why that is the case. So let us do some simplifications first of all. So the first so I wish I had a board, because if I had a board now, then I would just do the simplification. But so you will just have to do it in your mind. So let's assume there's no food. So the N is equal to 0. So this equation here, we take it out. So what that means is that there's no reason for the cells to go anywhere. The only two competing forces now is diffusion and attraction due to this self-interaction of the cells. And then the next simplification I will make is to take the alpha equal to 0. So the alpha equal to 0 is just to say, well, we're on time scales that are so short that we can just ignore this chemical degradation rates. And that's fine, right? You have to know in which setting that's fine, but you can do that in certain settings. And then finally, I will assume that this S is in quasi-equilibrium. And what that means is that I can just put a 0 here on the left-hand side. So just by saying that the time derivative is 0, I'm not saying that the S is independent of time, right? So the rho here, of course, depends on time. And because the rho depends on time, the solution to this equation is going to depend on time. But what this is saying is that immediately after a cell produces some chemoattractant, the chemoattractant equilibrates infinitely quickly, so that immediately this equation is in equilibrium. And so if you put a 0 here, then what you have left is the Laplacian of S and the rho term. And that is now just the Poisson equation, right? And this you know how to solve. I mean, this is like a basic equation in PDEs, one of the most famous equations in PDEs. And you have explicit solution for that. So you can write down the S in terms of the rho, and then you can plug the C in the first equation, and you basically reduce everything to just one equation. And so this is the simplified model that I show you up here. So I left out all the constants now, because, okay, the biologists, of course, they care about the constants. As a mathematician, you can allow yourself to just ignore the constants, do all the analysis, and then at the end, you put the constants back in, because the constants are not the difficult thing here, right? So I just left out all the constants so that we can concentrate on the structure of the equation. And so the diffusion is like before. And this term here is basically the log of x divided by 2 pi is the fundamental solution of the Laplacian in 2 dimensions. So in 2 dimensions, and here we're in R2, as you see, you can write down this equation like that, the first equation that you get. And now this equation has some really interesting properties. And this is called the critical mass phenomenon. So if you know what the mass of your cells are, like how many cells do you put, let's call the total mass M. And this M is independent of time, because here we're not considering cell division, we're not considering cell birth, so the number of cells is constant. They just move around. And of course, again, that's an assumption, and this only is true on very short time scales again. Or for certain types of cells. And okay, so this M is going to be just a constant, and we fix it at the beginning, right? So our initial distribution at time t equal to 0 has a certain mass. And now what is really interesting to look at is the second moment, or in particular, the evolution of the second moment, because the second moment tells you in a way how spread your distribution is. It's like your variance, right? And if you take the derivative, then it tells you if the second moment is increasing or decreasing. So are your cells spreading in space, or are they concentrating more and more on a point? And that's a very important basic information, because it tells you in the dynamics of the model which of these two forces is dominating. You know, is it the diffusion that is dominating with time, or is it the attraction that is dominating with time? And this is basically what this little calculation here gives us. And I encourage you to do this calculation. This is not too difficult. So you pull in the time derivative onto the raw, then you have dt raw here, then you plug in the equation, and then you have to do some integration by parts and sort of 2 or 3 lines of computation. And then you get this expression here on the right-hand side. And then you can immediately see something very interesting, which is that if you take m to be less than this 8 pi, of course, in real life, this is not going to be 8 pi. This is going to be involving all the constants that we had before, right? But here it's 8 pi. So then you don't have enough cells to really feel the attraction from the other cells. And so this, and as a result, the diffusion wins. The diffusion overcomes the self-attraction. And you can see this because here this right-hand side is then positive. And so that means that the evolution at the second moment always increases. So the cells keep spreading in space. And then if you have too many cells, the opposite happens. So this right-hand side is negative. And so the second moment decreases. And so the cells are concentrating more and more until the second moment hits 0, which means the raw is then just a Dirac. And that means you have reached a blow up in your equation, essentially. The equation breaks down. And this means that the self-attraction overwhelms diffusion because you have so many cells. And in between, there's this critical mass, 8 pi, where these two forces, the diffusion and the attraction, are exactly in balance. And there you can see that you have infinitely many stationary states for your equation, and that you have patterns that can emerge. And so the reason why this became so famous among mathematicians is because this is a very peculiar behavior for a PDE. So this PDE is a complicated PDE in the sense that it is nonlinear, because this term, for example, you have raw that appears twice in a product. It is nonlinear, and it's also non-local, because here this star is a convolution. So you're integrating over the whole space. And so the solution to this equation, so raw of Tx at the point x, is going to depend on the value of the solution raw at all the other points in space. And so this is what is meant by non-locality. And non-local and non-linear equations, they're usually very difficult to solve. And so, yeah, it's very interesting that the long-time behavior of this model, so what the long-time asymptotics, what happens as t goes to infinity, only depends on your choice of mass, essentially. It only depends how many cells you put. And that's, yeah, that's a very interesting behavior for a PDE. And so this has been studied a lot as a representative model for more complicated nonlinear, non-local PDEs that exhibit that kind of behavior. Okay, at this point, I should say that I'm very happy to take questions or be stopped. And you know, if you just want to clarify something or make a comment, feel free to just unmute yourself and interrupt me or put your questions in the chat. Okay, so this is not the whole story yet. I want to tell you about another experiment that you can do with these E. coli cells. So this is an experiment that has been done already by Adler in the 1960s. Now, this is a newer version of that experiment, but essentially you take a tube and then you put cells on one end, so here the left-hand side, and then you put some nutrient on the other end, and then the cells are going to start moving towards the nutrient. And if you do that, then you see that you have something like a moving front, like literally a traveling wave. And you see here the same tube at regular time shots. And this is the density profile. So you see that really this moving front is displacing at a constant speed. And the profiles itself it's asymmetric, right? But it looks very constant as it moves. So this looks like there should be a very beautiful equation describing this. But now if you go back to this model, the Keller-Segel model that is supposed to describe the behavior of these cells, you will notice that this equation does not allow for traveling wave solutions. There are no traveling wave solutions to this. So clearly there's something that cannot be true, right? Something here is not, the model is somehow not capturing one very important behavior or dynamic that we actually observe in experiments. And that has triggered a line of research where people said, well, maybe this model is too simplified or it's too macroscopic. And we actually have to go into the microscopic level and look at what happens at the interaction between each of the cells. And so for that, let me just briefly say how these cells actually move. So E. coli, they have these flagella on their outer membrane, and they move in two phases. So there's the run phase and there's the tumble phase. So in the run phase, they just rotate all their flagella in one direction and propel themselves forward like a little propeller. And then in the tumble phase, which is just very short, they put all the flagella to all sides, and then they randomly select a new direction and they go again. And so what you get is that the cells go sort of in a straight line at a constant speed. They tumble, so they change direction, and then they go off to another direction. And when I said that they're not able to control the new direction that they're moving to, you would wonder, then how are they able to get anywhere where they want to go to, right? If they cannot control the direction. Well, now we have understood how this works. And I find this very interesting that they're not able to control where they turn to, but they're able to control how often they turn. So the frequency of turning is controlled by the environment. So what is the concentration change in the chemoattractant and in the nutrient? And this actually explains how you can observe this very nice coordinated behavior on the macroscopic scale. Even though if you zoom in and you follow, so this here is a zoom in into this peak of the moving front, and you see all these lines here are the trajectories of the individual cells. So this is something that was not possible 25 years ago or in the 70s when this model was developed, right? But nowadays we're able to film a single cell using like green fluorescent proteins and get information really, how does each cell react to its environment? And so here you see the trajectory of a single cell. And you see that it's sometimes turning in the wrong direction, right? It's sometimes turning to run backwards. But in average, it's moving to the right. And the reason for that is because if it's going in a good direction, so if it's going to the right, then it just tumbles less often. So, and this is how you get this overall effect. Now, there is a model for this. So this model was proposed in the 80s by Ottmar Dunbar and Elt, and they said, well, we would like to describe this kind of tumbling behavior. And what I really like about this idea is that they took inspiration from physics, in particular from statistical mechanics. So those of you who know the Boltzmann equation describes how gas molecules move around, how they bump into each other. And here they're using exactly the same idea, the same type of model, but what they do is instead of gas particles, you now have cells. And gas particles, they just collide, and then incoming angle equal outcoming angle, and they go off in different directions. So here the collision is replaced by this tumbling kernel or tumbling frequency. So when two cells communicate via the chemoattractant, they then decide to change direction based on that interaction. They tumble and they change. So this is what this right-hand side is describing. So here we're not modeling anymore just raw of Tx. So raw of Tx is the macroscopic density. There we have no information about velocities. But if you want to describe the tumbling, so how, in which direction do the cells move, and then how do they decide to change that direction, you have to consider also the velocities. So this is why we are modeling now this bacterial density that also depends on the velocity. So this is why it's called kinetic, because there's this information on the velocity in here as well. Okay, the left-hand side is basically a transport term. So what this is saying is that the F is changing, or is being transported along a trajectory of a cell moving with velocity v. And then on the right-hand side, you have the change along this trajectory of the cell due to this tumbling event. So this T is a tumbling kernel that depends on the chemotractant, of course, that says what is the frequency of changing direction from V prime to V. And so what this is doing, it's basically saying, well, I'm here now summing up all the cells that have moved at some other direction V prime and that are now changing and that move to direction V. So this is basically like a source term. I'm adding in all these cells that were traveling in some other direction and now traveling sorry, into direction V. And then you have to also take out those cells that are traveling at direction V, and then they tumble, and then they have a different velocity. So that's the second term here. So it's like a source and a sink term. And this you can think of as a collision operator in physics or in kinetic theory. Okay, so if you have this equation, this is kind of a mesoscopic equation, because it's not modeling how the position of each of the particle changes of each of the cells. That would be really a particle model or a microscopic model. And it's also not the same macroscopic PDE that we saw at the beginning. It's in between. It's the kinetic scale. But we can now make the connection between this kinetic model and the PDE that I showed you, the Patlak-Keller-Segel model. And that's what I want to show you next. And for that, we need some assumptions. I haven't even told you what this T is. So we have to write down some expression for this tumbling kernel, right? And here we just make a very general assumption. We just say this is some function of the directional derivative. So we know that when the concentration of S decreases along the trajectory of a cell, then the cell will realize it is going in a bad direction because the S is decreasing, so it's going away from the group. So it wants to tumble more often. So the frequency will increase, right? So this is why this psi is a decreasing function. So the higher sort of the increase in the S, the less often the cells tumble. And you also see that it only depends on direction that the cell was going in, not the new direction. So this is someone changing direction from V prime to V. And now there's an assumption. And the assumption is that the influence of this chemotractant is only very small. So this psi zero here is a kind of basal tumbling frequency. So if there would be no chemotractant, no other cell, there's just one cell by itself, all alone in the universe, it would still tumble. And it would tumble at this basic rate psi zero. And now if you add other cells and other influences, it will either increase or decrease its tumbling frequency based on that. And that influence is small. So this is what this is saying. And now what you can also do is you can rescale your equation. I've written it here in a more compact form, and you have to choose a certain type of scaling between the time and the space. And then you get the epsilon appearing in these different places here. And the next thing you want to do is take the epsilon to zero. So this is a whole, this could be a whole lecture course just doing that. I mean, this diffusive limit. But I will skip that and just say that this is a kind of mathematical way of zooming out, right? So you're at the kinetic level and you want to now zoom out and get to the macroscopic level, back to the PDE we had before. So I'm going to take epsilon to 0. And what you get in the limit is something that looks like this. And that's quite remarkable because you will, if you remember the equation I showed you, this looks very similar. Actually, it looks exactly the same, with the only difference that here, instead of the gradient of S and the gradient of N, I have these functions U now. And these functions U are given by this complicated expression that depend on the phi. So the phi, if I go back one slide, was this function here that tells you the bias of the basic tumbling frequency. And okay, you know, this phi is something that you would want to estimate from experiments, and there are different guesses of what this phi could be in practice. But clearly, this is not the same as the gradient of S. And here's a very fundamental insight. And basically, this Pattler-Killer-Segel model is nothing but a linearization of what is actually going on. So if you take this US and this UN and you linearize for the phi, then you get back to the gradient. And that's actually really, yeah, this explains a lot. And now here also this expression, this U of S is bounded. Of course, it's more nonlinear because this is more complicated, but also this model has traveling wave solutions. So here you can now explicitly go and show that for certain choices of phi that are estimated and that are consistent with the biological experiments, you can actually solve this and you can show that you can recover traveling wave solutions. And I want to show you this picture, which is very nice. So the black lines are numerical solutions to the model, to this new model that was derived in this diffusive limit. And then the colored dots are experiments. So this is the same front moving, and this comes from the shot that I showed you before, this picture here. This is basically a comparison of this picture with numerical simulations at 3 different time shots. And you see these tails here. I mean, these are probably dead cells because the model is not taking cell death into account, as I said. So there's a bit of discrepancy here, but this is a remarkable fit for numerical simulations compared to a model. And this is with the phi being minus the sine of y. And so in that case, you get the U of S to be the sine function. And this is actually now the equation that we looked at. And I realized that I'm running out of time and that it's getting late. So I think I will just quickly jump to sort of the main statement of what we proved, because I think it's nicer that I spend some time sort of explaining the setup and the context. But this is the equation that we looked at. So we looked at exactly the setting where you have this nonlinear drift term with the sine, which is also discontinuous because the sine function is discontinuous. You have n equal to 0, so you consider there's no food around, and you just look at sort of the competition between the attraction and the diffusion. And you also consider the S to be in quasi-equilibrium, as we did before with the simplified Killer-Segel model. And you see that there's no time derivative here for the S. But now this is not anymore the Poisson equation, right? So you can still solve it. I mean, S in terms of rho, but it will be a bit more complicated and would have the alpha in it. And then what we want to show is a stability estimate like this. So the rho infinity here is the solution to this equation after very long times. So you want to understand how does, you know, what happens after long times. And the rho 0 is your initial distribution. And what you're saying is that if initially you're already close to the equilibrium, then you can show that you converge to the equilibrium with an exponential rate, where this gamma can be written explicitly in the parameters of the model. So this is a very nice stability estimate. And the fact that we have this discontinuous nonlinearity here is something that makes the analysis very difficult. But what is nice that you can actually find the stationary states explicitly. So you can write down what they are. It's like a whole family of just exponential functions like this. And then what we're trying to prove is this local stability for the raw infinity. And let me just tell you sort of the key idea to get this result is that we split the proof into considering the shape, the evolution of the shape. So how does the profile change from the evolution of the center of the profile? So where is it centered, right? How does it move around in space? So we try to separate these two. Of course, they're dependent on each other. So you cannot have them as independent equations, but you can analyze them independently. And to do that, you define this x of t, which is the maximum of the s. So if the blue curve here is the s, right? This x of t here would be the maximum. Then the orange curve or brown curve here is the derivative of s. And then you take the sine of this. So the green is then what you would be actually plugging into your model, the sine of the dxs. So this is what appears here, right? And if alpha is equal to 0, there's actually analysis that you can do quite easily. But if you have alpha greater than 0 and the sine function here, then things are very complicated. But you can derive this equation x dot that tells you how the position of the peak changes. And so you can analyze this equation now separately from how you analyze the motion of the profile. And then there's like a couple of reformulations that you can do for the model itself. So you can put yourself in a moving frame around this peak. And then the shape is given by this equation, and the center evolves according to its own little ODE. And this is the theorem that I just wanted to show you to kind of conclude. So now this is the fully like detailed mathematical statement of this informal goal that I stated before. So we show that if initially you're close to the equilibrium state, then you have this convergence with the rates. And the norm that you use here is this H1, this weighted H1 norm, which is a norm that is weighted by the stationary state itself and with this exponential weight that depends on chi. So chi is the chemotractant force, and it contains a sort of L2 term, and then it contains the L2 norm of the derivative. So this is why we called it H1. And you have an explicit bound on the rate of convergence in terms of the parameters of the model. And there's a lot more I could say about this, but I think I should sort of get to an end so that we have some time to discuss as well. So let me jump through all the proofs and just go to the end to say that, well, thanks a lot for your attention. And there's a lot of fun things you can do with E. coli. So for example, this is a San Diego Beach scene that is drawn with a living bacteria, where each of these cells has been injected with a different type of fluorescent protein. I mean, this idea of coloring bacteria this way actually won the Nobel Prize, I think, was it in chemistry, for the idea of using these fluorescent proteins for science experiments in the sense of, for example, filming these cells, which was not possible before. So yeah, I thought this was a fun little piece of bacterial artwork at the end, and let me finish with that.
Thank you very much, Miss. Any question for any participant? ¿Alguna pregunta hacia la profesora?
Yo comprendo un poquito español, pero no. Not very much, but yes, if anybody has questions in Spanish, that's also fine.
Yeah, well, hay una pregunta. Well, one question about the produce or the reproductions or production chemistry reaction about the cell how growing about the cells,
Sorry?
more or less.
How?
The question is that how meaning about the reaction of the cells?
The reaction time, for example?
Yeah,
so how
the reaction time about the shape of the center, these steps, how meaning, or what is the biological meaning is that?
So in general, for the model, yeah, you're right. So in real life, the cell will need, so the cells are not able to measure instantaneously the changes in concentration. So they're not actually measuring the gradient at a point, but rather they compare sort of the number of bound receptors on the outer membrane with, I don't remember exactly what the numbers are, like 1 millisecond ago with 5 milliseconds ago or something like that. So there is a certain delay, right? Like once they have reached a region where the S is very small suddenly, they will only notice that a little bit later. And there's a separate sort of investigation that you can do where you include that into the model. So where you incorporate into the model this time delay, and this explains the kind of plateauing that you might see on that peak. I don't know if that answers the question, but it's an effect that is there that I ignored here because we understand it and because it's relatively small. But was that the question?
Yeah. And one question about me is about the delay, about the time, less instantly. How is the impact about the impulsive differential equation about PDE? Probably in this best way, or what's the distributional differential equation about the delay is about the delay differential partial equation. About the impulsive is better, probably, in this consideration.
You can build the delay into the term that contains the chemotractant S. So I don't remember now exactly how the equation goes, but you can basically say, well, there is a certain time that the cell needs to react to a change in concentration. You can call that tau. And now you have a T and a tau in your equation, and then you try to see what is the effect of that tau.
I know, understand. It's better for the analysis. Well, another question, alguna pregunta.
I think someone is raising their hand.
Juan Carlos.
Hi.
Hello.
Hello. I was kind of late, but I got curious about how you made bacteria draw that picture you showed at the end. I mean,
okay,
how
yeah,
did you
I can tell it.
Get bacteria to go in the direction you wanted, right? So that they made that picture.
I mean, I have not drawn the picture myself, so I should not comment on the details of that because I don't know exactly how they did the picture, right? I mean, they could have just placed the cells there and take the picture immediately, right? Not that the cells moved to those locations themselves, I assume. But in terms of the this method of filming cells, so this picture that I showed here, for example, is produced with this method, all these trajectory of cells. what they do is they take this protein, which I think usually is in a certain jellyfish. They extract this protein and then inject it into the cell. And so the cell will reproduce this protein, and it's fluorescent protein. So if you shine an ultraviolet lights on the cell, then it will start to glow in green or in some other color. And so this will allow you to sort of film a single cell in a group of cells. And so this idea of using this green fluorescent protein in this way, this is what received the Nobel Prize.
Okay, thank you. Thank you, teacher.
Another question is, what is the adventure about this work in the biology?
So I think here this model is still in a quite idealized setting in a way. So I guess that often the processes that biologists are looking to model might be much more complex than that. And also you can make this model more complex. You can also take, for example, internal pathways into account. You know, there's lots of interesting things happening inside the cell. And you might try to link that with this model, how each cell is reacting with its environment. So there's many versions of this model that combine with other models and are more complex, and therefore then maybe more directly applicable. But I think, as I said, this Keller-Segal model has gained a lot of popularity within the mathematics community for a reason that maybe for the biologists is not so interesting, which is this critical mass phenomenon, which is mathematically a very peculiar behavior of a PDE, that if you understand it, you can also understand the behavior of other type of reaction diffusion equations. So what I'm saying is that the kind of questions that biologists and mathematicians are trying to answer using this model or using modeling in general are probably quite different. But then still there's a lot of questions also where mathematicians can help. Like, for example, the fact that you can, by estimating the number of cells, you actually can predict what the whole group of cells is doing, whether it's getting closer together or spreading around. I mean, simple questions like this on the dynamics.
Another question is, what book can I read about this topic, about the mathematics and the biology?
Oh, there's many. There's like a couple of really good general reviews. And there's one by Alt. Alt was in the 60s. There's Ottmer. Ottmer has a couple of review. I mean, if there's someone who's specifically interested in this, I can send some references afterwards. But there's a couple of really nice sort of overview papers that summarize what is known, what all the different models do. There's also a nice book by Boudrin and Burke on pattern formations in general. And there's also a whole section on these bacterial patterns that are due to bacterial chemotaxis.
Thank you. Another question. I think no more question. Right. Well, thank you for the talk. It's very nicely about the dynamic, about the material. And thank you very much, Miss, for the talk. And thank you again for accepting to share this this topic about interesting about the biology. It's so complicated, some topic about me. Yeah, no more question. Well, thank you very much about the talk. In another opportunity, I hope to share another topic about job or experience about the simple school with I meet with you. Thank you.
Yeah, thanks a lot for the invitation. It was really a pleasure.
And thank you very much. Gracias a los participantes de hoy día que nos han acompañado. También en la siguiente semana tenemos el tema de ecuaciones diferenciales parciales también aplicado a cinemática, un aspecto más físico. El tema de la siguiente semana. Y nuevamente gracias, profesora, por la charla. Bueno, en español, disculpe. Y nuevamente, thank you very much. I know it's very late in Bonn. Thank you for the passion about the topic and the talk, and very nicely.
Thank you. Greetings to all of you from Bonn.
Ciao, Miss. See you, Miss.